Welcome to the Spring 2024 NFT Hydroponics experiment, performed in Dr. Ferrarizi's hydroponics greenhouse by Nathan Fennell. This experiment studied the effects of supplemental light and varying pH levels on leafy greens within a nutrient film technique, or NFT, hydroponics system. NFT systems are a water culture-based growing system where plants are suspended in hydroconductive growing media and subjected to a constant recirculating flow of nutrient solution. The process began with assembling the NFT system, then hanging the prize element top light PPR lighting fixture. 20 gallons of nutrient solution were then injected at a concentration of 100 ppm to each culture using Jack's FEED 15520 fertilizer. This was all followed by the delicate process of transplanting each lettuce plant into their new hydroponic home, as you can see here. This system had 9 separate cultures kept at 3 pH levels, each nourishing 24 plants, totaling 216 subjects. 3 plants from each experimental group were studied, totaling 54 data points. Every week, data was collected on water usage, pH, water temperature, and plant size. Data was also collected at the end of each season on chlorophyll and anthocyanin content. pH and water level were adjusted weekly in hopes of ensuring that the experimental cultures stayed within their intended zones. Now, let's talk about some results. It quickly became apparent that the plants did not appreciate the over 1,000 micromoles per meter squared of supplemental light that they were bombarded with 24 hours a day, 7 days a week. The supplemental plants on both seasons had severely stunted growth, thick, leathery leaves, and were about 30 to 40% smaller in each dimension than the sunlight plants. In the first season, the Rockeye oak leaf lettuce cultivar already contained some reg pigment from the anthocyanin that was naturally present in its genetics, speckling the sunlight plants, but overwhelming the supplemental plants. Due to the intense amount of blue light on the supplementally lit plants, they turned completely dark red and had extremely high anthocyanin levels. The second season crop was a pure green lettuce cultivar from Rex called butter leaf lettuce. The supplemental lighting group stayed green throughout the season this time, turning a pale green as they became stressed and began to dry out and die from the excessive lighting. In both seasons, the supplemental lighting plants were so stressed that they became prone to bacterial and fungal infections. This proved to be deadly for more than half of the first season plants, with a bacteria infection running rampant during the last week before harvest, rendering most of them inedible. The sunlight plants, on the other hand, were completely immune during both seasons. Sometimes, natural is just better. Overall, this NFT bench worked perfectly during both seasons, serving as a reliable testing area for supplemental lighting and pH levels. Water flow and distribution were sufficient, pumps were reliable, and the natural light levels of the greenhouse proved to be just perfect for the leafy greens. However, there were a few problems in this experiment. The cultures did not respond reliably or consistently to the pH adjustments, or so it seemed from the data collected. pH was adjusted to the experimental levels of 4.6, 6.1, and 7.6 once per week, but all of the groups seemed to quickly return to within 0.4 pH of each other by the time the data was collected the next week. This was due to a faulty data collection sequence. Water levels were restored before the pH readings were taken, diluting the true pH reading of the culture that week by almost 10 gallons. In the next NFT hydroponics experiment, the way that the pH is measured and adjusted, as well as the regularity of the measurements and adjustments, must be changed. It is apparent that maintaining an accurate pH for an experiment like this requires a rigorous maintenance routine. And, just for fun, the lab also grew other crops, such as cucumber, tomatoes, and peppers in the greenhouse. These crops were also grown hydroponically using the Dutch bucket method. Later in the season, they made for a refreshing snack break while painstakingly measuring 54 different heads of lettuce in 3 different dimensions. From this experiment, we can conclude that too much light not only wastes energy, but can radically alter plant morphology and immunity in negative ways. This experiment, in the end, is a testimony as to why researching and testing light tolerance and efficiency for a specific cultivar are key before purchasing a supplemental lighting system. Thank you very much for watching, and I hope that this video can benefit your future hydroponic ventures. Happy growing from Nathan Fennell and the Hydroponics Club at UGA.